This is the first of our roundtable discussions. The topic is what clinical and research questions should be a priority for multimodal neuroimaging. We've gotten quite a sampling from today. As you can tell, I'm losing my voice, so I'm going to make this very short and sweet. There are 2 or 3 microphones up there that each of the speakers have. Anyone from the audience is welcome to approach the solo microphone in the middle and sort of kick it off. Does anyone have a starting point? There's an on-off switch on the back.
Hello, hello. Siri, turn the mic on. So we were actually asked as speakers to come up with some sort of clinical application, and I came in talking about epilepsy, and I'm going to say this is a hugely important disease, worthy of study, and interesting from the question of multimodal imaging, because unimodal imaging is such a abject failure in this disease. So I'll put that right out there, and then I'm going to make a play for the thing which it has my passion these days, and which is underfunded and underappreciated in the NIH in a huge way. And let me just start off by saying that the NIH guidelines seem to favor the idea of making living zombies. And there's really very, very little attention paid to the question of human consciousness within what we do as a research question at the NIH. And I would claim that having a heart beating that's feeding a brain that's not thinking is a waste of everything. And I would like very much to see increased attention placed on the study of human consciousness. And frankly, I can think of no better area in which multimodal imaging, multimodal data acquisition is clearly essential. Clearly, we have no tools right now which are reliable indicators using any kind of instrumented acquisition for doing these kinds of studies. So, you know, the very notion of being able to understand whether somebody's inside this box is, some human is inside this box of me, that's one that's worthy of study.
That's a really important question, and one also wonders whether that person's conscious when they're lying in the intensive care unit, or whether they're in a profound state of status epilepticus, where now they're in a state where you can't see the overt seizures, and that's a really life-threatening situation for someone who can make a recovery. So it's a really, really important thing. So I just wanted to add.
Yeah, I wanted to co-sign your intent to raise the issues about levels of consciousness, minimally consciousness, coma, deep consciousness, and so on. One of the questions that patients and families have in these situations is my loved one going to recover? And the question of the duration of the disorder, particularly in the case of traumatic brain injury and so on, is probably an answerable question if we just knew how to approach it. The questions the way that we approach it now with imaging and asking people to follow commands and so on in the scanner don't really do it. But if we were smart and combined perhaps EEG, perhaps deep brain stimulation, perhaps other computational methods, if we really focused on it, I believe it's an answerable question. So I really applaud you for raising this issue.
It is a funny priority I would like to see addressed at some level.
Just wanted to co-sign as well, and I think it's a super important point, and we've shown that resting state, baseline resting state connectivity, like with the the baseline anticorrelations can predict who will come out of a coma, but we really need multimodal imaging to really up that prediction. And I'll just take this opportunity to say, I think the biggest, one of the biggest things is earlier identification and prediction, multimodal prediction of future outcome to really help the society right now. I think that's the biggest thing we could do.
Perhaps something more methodological to link to this. With EEG, we can do long-term recordings. With MRI, we can't really do long-term recordings. Like with EEG, we can record for days, or at least like for a night. It's not too difficult. For days or even up to weeks is technically possible. Whereas with fMRI, we basically limit to 1 hour, 2 hours max. There's also other modalities that have not been discussed today, but we have at Radboud University in Nijmegen, we have a project, Healthy Brain Project, in which we're acquiring data from healthy individuals, which also includes motion tracking data while they're walking around. So we now have, from some patients, we have about a year of motion tracking data, very detailed motion tracking data, it's like it's with a wearable device, but that's sampling much faster. And last week, when discussing with the data stewards, I realized that this is about 100 gigabytes of data that's recorded per subject. And there's a lot of detailed information in there, which I think at this moment we're not yet considering in great detail in the analysis, is that there's other modalities, behavioral modalities that we can record, that we can track. Video is something that we could also track for long periods of time, also very large, very difficult to analyze. But it falls in a very different category than the types of signals that the most of us are used to looking at, which are signals that we record from the brain rather than from the behavior.
Yeah, to link to what Robert said with what Mark said, my sort of thing that I'd advocate for is let's put the brain back in the body. I mean, we've forgotten, here it is in this dish that we call the fMRI scanner, or we just care about the EEG. In how many cases have we seen, if you're in the ICU, with someone who's perhaps being monitored for being, or checked for being brain dead? Are we monitoring other signals from their bodies? We don't do that. Okay, so they've got EKG. But there are plenty of other things that we could be doing in people who are unconscious, as there are in people who are healthy. We should be sampling the body as well as the brain and putting the activity of the body into the context of the brain activity.
Maybe coming back to, you started from epilepsy, and for me you have many different ways of being in the ICU. Epilepsy is one pathology, so maybe can you comment a bit more specifically what you have in mind with epilepsy? What is specific to epilepsy? Is it just one use case where you wouldn't have these types of questions, or?
So in the context of this meeting, of discussing multimodal approaches to data acquisition, I think that the diseases like this, which have been the most resistant to traditional diagnostics, are the ones in which we probably need to address the question of whether multiple diagnostic avenues are going to be the way to access the disease. It's back to this whole theme of the whole world in machine learning of weak predictors combining to produce strong predictions. And every one of the tools that we have in this disease, and there are probably others, I'm not expert in anything, let alone all the diseases, but you have weak signals on the PET, you have weak signals in the EEG, you have weak signals in SPECT, you have weak signals in like almost anything you could use in this clinical condition. And the organized process of synthesizing and sharing that information, I'll defer to Vince. The organized process of really combining information for incommensurate modalities is what we need.
Someone's really a geek when they draw circuit diagrams and they doodle on their paper. So, what'd you design there? A new piece of equipment here for us? Yeah, he's got a new data collection modality for us. Yes. I think I totally agree with everything that's been said, and also I was going to say the same thing about sort of the embodied brain type concept, which is that we've got a bunch of other sources of data, and it's not just about the brain, right? And there actually are feedback loops and all sorts of things that we're kind of missing if we ignore it. But I would also say that in terms of just, clinical applications, I think, I don't think we have exactly the solution yet, but multimodal imaging, and then we need better unsupervised learning approaches, basically, because right now we need an answer to predict something, right? And we're assuming that answer ahead of time, and we know it's wrong, So it's this vicious circle, and we can't break out of it. It's hard to break out of it, and so there has been some progress in terms of just trying to let the data sort of inform existing clinical categories and revise them and things like that. But that, I think, is an area that really needs to be pushed a lot further. And I think in the context of multimodal data, that can really help, because you have these blind spots where see things will split up differently depending on different sources of data that are missing. And I think we're kind of poised to make some advances if we can devote some resources there. So.
I think that's very true, and I think also to the question of why epilepsy, for example, and then also why states of consciousness, is that that's a disorder that's really characterized by spontaneous events, and so it becomes something that's very hard to understand without having kind of multiple different measures unfolding at the same time. I think it's the same for consciousness. I mean, there's the really critical, important problem of people with disrupted states of consciousness, but even our natural states of consciousness as we're awake and asleep, as Katie's talk really showed, these are such spontaneous and dynamic fluctuations that they really benefit, I think, from that added simultaneous information from different modalities as well.
I'd also like to add another area that I think is a very important area that could benefit a long ways with multimodal imaging, and that is the area of social neuroscience, the area of studying interpersonal interactions, particularly interpersonal interactions that happen in live, spontaneous types of situations. It's a little bit self-serving because, of course, this is what I'm going to talk about on Friday, but I think that it's a new area that challenges many imaging modalities and the possibility of combining them together to answer questions about the underlying neurophysiological substrates that are actually instrumental in autism and other developmental disorders. It may provide incredible opportunities for us if we begin to think about combinatorial approaches to this area. So I'd just like to add this to the list of possible priorities for multimodal imaging.
I just want to pick up on something that Vince mentioned about, letting the data drive things a bit more. And it sort of strikes me a little bit that epilepsy is probably an exception because there's a very good classification of seizures, and there has been by the International League Against Epilepsy for decades. But there are other things, the neuropsychiatric disorders, for instance, that are a mess. There's this umbrella term, schizophrenia. That's an umbrella term. And so, no wonder people aren't getting it anywhere. So let's rethink how we look at patients. And right, and as a group. So what, exactly? And so I'm thinking about the approaches like people like Lucina Udin are using in Florida, where she is looking at a brain region, and she's a fan of the insula. And so she's tapping, abnormal kind of connections and functions in the insula, and looking at, has identified 3 disorders that occur in 3 different parts of the lifespan that seem to show common features and involve the insula. And they're quite different. I mean, we're talking about frontotemporal dementia on the old side of the lifespan, and then things like autism on the other side. So that's a very different way of looking at it.
I think I'd like to propose a slightly different application. I'm not really sure I would call it a pathology or a disease, but childhood and pediatrics. So I shy away from using MRI, or functional MRI anyway, with children under the age of 7 or 8, because it's hard. And there are certain modalities that are really natural for younger children. And then when we get to infants, maybe MRI is okay again. But the different modalities have different strengths at different periods of development, and also depending on what your questions are. And putting in a bunch of, looking at human movement in infants is incredibly informative of just characterizing that. So I think because we can't use a single modality to really cover all of the territory we want to cover from birth until adolescence, I think that whole category of research question or clinical question is really well served by multimodal imaging. So it's also the time when we could do the most good. So I would propose that.
So another thing I'd like to consider in this context is whether the combination of, PO2 and respiration and CO2 and EMG and galvanic skin response and maybe EEG, a number of incredibly cheap modalities, has the possibility to equal one fMRI. And this is something that clinically we really truly need. We are in a space where electronics are cheap. You can build, a massive computer on a credit card size object, and we should be considering whether this is an opportunity to step away from these outrageously expensive things that we do, and to use the data combination techniques which are growing in multimodal data sets in order to try and augment the clinical practice with something inexpensive.
Yeah, and I think that incorporating economics and cost in terms of modality, I think, is important in the analysis approaches too. Comparing all these things and trying to say, well, if I'm trying to predict some outcome, I can do just as well with this combination of modalities versus that one in many cases. And I think that's something that's quite easy to do these days, given the data, right? And so I think, I completely agree with that.
And I have a slightly different take on the clinical multimodal. So if someone came into you're trying to diagnose someone comes into your office and so on, you tend to be very time limited, right? You maybe have a half hour to spend on this person to collect data. You're not going to collect a lifetime of fMRI to understand the disease of the population. You can't collect that much data. And so one of the things that I think about for multimodal is how to collect the most amount of data in the shortest period of time in terms of putting a whole bunch, so in other words, could I say put on near-infrared spectroscopy and EEG and do in 10 minutes maybe what it would have taken me 30 or an hour to do in terms of number of trials. And being able to take advantage of the fact that these are concurrent measurements such that you start to look at the noise and it fluctuates from trial to trial, and if you see it in both modalities, it was brain related, it wasn't instrument related. And so we've been trying, in my lab in particular, we've been trying to come up with ways to use that kind of concurrent multimodal information to infer something more about the brain and the fluctuations within the brain independent of the modalities, as a way of kind of getting away from the need to collect massive amounts of data to understand something about the brain.
That also reminds me of, just going back to MR, MR fingerprinting and things like that, because I think there's a lot more that can be tapped into there in the functional domain as well, in terms of just density of information, right? So that would be, that's obviously expensive to get somebody in there, but still, I think the same kind of theme could work.
Different question. Do you think we run the risk by collecting a lot of data like multimodal, maybe even in cases where we don't have clear hypotheses, and let the data drive us to lose, to stray apart from hypothesis-driven research? Is there danger to that, especially maybe not in people that, just want to use a lot of methods. If they don't start with a question, but rather with, I want to do fMRI and EEG and that, where's the danger in that, or is there danger in that?
So I see that risk, also because I'm in a more cognitive research setting where hypothesis-driven research is more important, and I will be addressing this tomorrow morning in my presentation.
Maybe also to jump on this, Vince, you said, okay, we need to, I mean, how do you quantify that you make a progress, right? You use these cheap devices, that's nice, but basically then how do you say these devices better than fMRI? And you said just before we should do unsupervised learning. And for me, there's kind of a mismatch of, I mean, how can you claim that you're better if you're only unsupervised? That's fun. And for me, validation of unsupervised model is still an unsolved problem, and I don't see how you can fix this. And otherwise, in order to validate and quantify, you need a clear target. So you need to do supervised learning. And basically, in order to start to accept or consider answering this question about, I mean, should I use this device or this modality, then you need to know what question and what are you trying to predict, and will you be able to predict reasonably well given the noisy labels, and so do you still consider that unsupervised learning is the way to go?
It's on. I still do. I think that the validation is going to play out in terms of actually does it predict and does it help people long term, right? And those studies will have to be done. So, for example, and I don't have the answer. I mean, but you know, if you basically use unsupervised learning to identify some new categories, you know, across diagnostic boundaries, and those are better able to predict future outcomes, then you know that process will help you learn. I think you don't stay unsupervised, right? You start unsupervised. It's a circle, right? I think it's always a circle where you kind of have. You have to have both. You have to have the hypothesis, you have to have the data-driven, and they both have to be humble, right? And inform one another. And so the validation is essential, but you know, maybe you broaden your horizons initially. You let the data tell you what might work. Try that out over time. Have some well-defined clinical questions that you're trying to predict or answer. Maybe it takes a few years to run a study and see how well that works prospectively.
So science did not start when the t-test was invented. And I think that we should all have much, much more respect for exploratory observation than we seem to these days. I'll just say it. I look at a journal like NeuroImage, and it seems to be all modifications of how I can change the 5th decimal place on some sort of statistical test. And this is just not the way science in the real world is done. Karl Popper is not in danger of telling me I'm wrong, but you know, real people don't walk around and form a hypothesis and test it every time they do a science experiment. And if I want to know whether the muscle in a frog's leg is different than the muscle in a human leg, I have to already have come to the conclusion that there are things like frogs and humans, and it doesn't require me to do a t-test, right? It requires skilled observation. And you know, tools like unsupervised learning, they are hypothesis generating tools, but you need them. You need data to form a hypothesis.
Okay, I'm having flashbacks here to NIH study section when I chaired it. And I'm not going to say anything about NeuroImage, even though I'm an editor for NeuroImage, so I'm not even going there. But just thinking back to just on grant review panels, I think we need to think a little bit more broadly, because that's where I think people need to have that balance. We couldn't, if we didn't have the odd fishing expedition and exploratory work, which was truly innovative, we wouldn't make progress in science. There has to be a balance, and we have to have some work that's innovative and a fishing expedition, and this other stuff has to be hypothesis driven, because we're never going to make progress. No new area can exist if we don't fish for it. And maybe that's controversial. Well, good. Let's have an argument here. But I think we need to seed new work.
Yeah, and I would also say that those don't have to be polar opposites either, right? So there's a whole spectrum in between, right, of more constrained and less constrained, and all of those can play a role.
I think we need to think more about how to make that argument palatable. I'm just coming from a study section on training grants, the F-level training grants. And the number of students that didn't get their grants funded because their work wasn't hypothesis-driven is hurtful. And I think partly it's not because the work wasn't considered valuable, it was that it wasn't phrased in a way that allowed the people reviewing the grants to actually endorse an exploratory method. So I think not only do we need to think about how much we value exploratory processes, we need to think about how to convince others, or how to write those paragraphs that embed it into our mainstream of thinking.
I'm just going to hop in and say there are a lot of program officers who are Skyped in or WebExed into this. And I know I've talked to many of you, I've talked to many people at conferences about the difficulties in funding and how sort of requests for funding work. Do any of you have suggestions of some kind of mechanism for funding this mixture of sort of fishing or more exploratory type things, but also with the funding caps the way that they are, how could we really get at these more expensive multimodal things that, because you're involved in more technologies, oftentimes there's startup costs in getting involved. If we start saying, hey, we're going to start giving out grants for multimodal imaging, suddenly everyone needs to be able to afford a NIR system or an EEG system or some other system to go with what they already have. Do you guys have thoughts on that?
I've got a thought just about, not so much that, but about writing program officers for thinking about things. I don't think that that's the problems at that end. The problem's the reviewing of grants. There's a mechanism called R21. It's for high-risk research. That's what this mechanism is exactly for, the kind of things that I'm talking about. But do reviewers do that in a study section? Do they have their R01 hats on? And they're reviewing those R21s with an R01 mentality. And that's a real problem, and I don't know how to fix that. And I think that's what's killing us.
You know, all grant applications come with guidelines, and the reviewers see those guidelines. And one of the guidelines can simply be that the NIH recognizes the importance of hypothesis-generating exploratory work. And you can simply turn around and say, this is that aspect of these proposal requests to which I'm responding. I'm doing exploratory work because we need to be able to get data in order to form ideas.
And just getting back to the actual writing of the proposals, I think there's also sort of a real key role that, and you had mentioned earlier, skilled observation, right? So skilled observation and just being insightful about what you, and communicating about what you're trying to do, right? I think that I see, as someone who's kind of at the intersection of application and sort of analytics and machine learning, I see a whole bunch of really cool analytic approaches, most of which will never get used. There's maybe a paper published, and then it just goes away because there's been almost no attention paid to what does it mean, why is it interesting, what are we trying to do here? It's more just, I got some data, I throw this cool algorithm at it, and I get something out, and I just kind of write something around it, right? So I think that's not everybody, but I think there is a whole, sort of in this interdisciplinary space, there's still, silos. People are still kind of siloed in their own camps, and I think that's one of the reasons why proposals that come through are missing. They're missing that part of things.
So about skillful observation, I really like that term because I'm still one of the people that thinks one can learn a lot from an N = 20 study if done properly. So I was really curious about your comments on that. You know, the hype is really about big data sets and so on, and there's definitely value in that. I'm not saying, but Lorenz, I think, said once that it's better to study one animal for 1,000 hours than 1,000 animals for each an hour. So what's your take on that?
Well, I'll make a brief comment, which is these days it's great. We've got lots of data, lots of things we can do with it, but you can also do a lot of very silly things really, really fast, right? And it can get you something that you can talk about, but if you don't go back and see what's under the hood, you're missing, you know, a piece of that. And I think we don't yet have all the tools we need to be able to do both, right? Because they're also, again, with just as with other things, there's a spectrum here, right? And so as you scale up your analysis, you kind of get further and further away from just looking at what's going on, and that's a risk, right? And so yeah, I mean that resonates with me, and I think that's an issue. I also think that large data doesn't solve all the problems. I think it solves some problems, and it helps you identify patterns that you can go back and look. But there are certain questions that require individual. And I still remember, you know, the early fMRI days, right? Doing a motor tapping task, one subject, okay, that's pretty obvious. There's something there. And no one's going to tell me that that's a meaningless data set, right? So other things are very subtle, and maybe you can't see it, you know, in one data set. But it really, so it kind of depends on the effect size as well. But,
yeah, I mean, most recently when I've been gathering these really long scans that have all the EEG, physiology, it's sort of like we have these really like deep data sets in a very small number of people. And one thing I really love about that is being able to really look so carefully at like one data set, and that even before trying to apply like some tools, I just look at all the time series, like how do things correlate, everything. You can get like a really interesting sense of like how much relationships there are between things in the data set. And I think that's really cool to then maybe try to scale up. Well, then I guess the trade-off is you don't know how much that's specific to the one person you're looking at, or if it's like generalizes, because sometimes I think, wow, I found this really interesting thing, and I'm like, I don't see it in any of my other subjects, so to what degree that's that generalized, but I think that does really help to try to generate different hypotheses that you can then maybe then bring in larger amounts of subjects, then you have a more targeted hypothesis of what's going on, whereas in the beginning I was just plotting signals like and looking at them for like hours, so without maybe a clear hypothesis.
I think the other thing is the open science movement has really pushed us to show more of the data. And I think showing more measures of variability instead of just having these histograms with means and standard deviations, but showing all of the observations. And people are coming up with beautiful ways to plot the data, informative ways that are very, very meaningful. I think that's what gives me a lot of hope for the future, because the way people are visualising and displaying data and helping people, informing them about their data set in a way that we haven't done before. And I really like that.
We're straying a bit from multimodal in these questions.
I just wanted to add to your comment about the small sample size. I mean, patients come in n's of one. And when the people that study epilepsy, for example, I mean, how many times you put a single patient in the scanner and you look for a single person's etiology with respect to the locus of the seizure and so on. And so the N of 1 study, the classical clinical case study, is an important part of our science. It's an important part of our foundation. And I don't want to lose that because of all the benefits we have from the large sample size studies, which are relatively new relative to the N of 1 study. I mean, remember Wernicke, remember Broca. I mean, those N of 1 studies have had an incredible impact on us. And so I don't want to lose that as we begin to think about more sophisticated approaches that require multimodal data sets and understanding of our variants.
That's really interesting. And in light of that, I'll revise a little bit I said before too, which is that like even though I may sometimes worry that something won't generalize to a lot of other people, maybe that's what the interesting thing is. Maybe it's that outlier that's going to tell us something really new that we can follow up on. So yeah, it's really interesting.
Maybe one comment too, following on Aina on what you said about the open science and the open data. I think what's hopefully changing is that people start to validate across data sets and across problems. I think what has been kind of a failure of the community is to have one new method on one private data set. And that's why you see also so much noise in the literature. And I think we need to fight this. And the way that we have more open data is also a way to kind of enforce authors to validate at a bigger scale with a barrier that is hopefully lower than what it used to be. So for me, this is the huge impact in terms of what this could bring. So for us to do better science that is more validated or...
Just for fun, I'll try to stray back to the multimodal imaging question. And I think it would be extremely instructive for our community to have clinical input on what are the most difficult diagnostic challenges for clinicians given the tools that currently exist. And that, those are the ones that are at least ripe for considering whether this data combinatorics thing is offering us complementary information. And, I'm not a clinician. I'm interested in epilepsy, but I could probably name, if I really pushed it, a couple of other disorders which share this feature that the clinicians, they barely know how to treat and they can't diagnose. And, we need to be putting our efforts there.
And to remain in the clinical sphere, Andreas and I were having a discussion at coffee time about clinicians maybe having to also change the way they're doing things. And Andreas's elegant work is a beautiful example of where the clinician is the rate-limiting step, and their ability to insert those electrodes and the visualization methods, that largely determines the success of what's going to happen to the patient, yet they're hampered by existing methods, whether they be their surgical technique or ways of actually visualizing their surgical targets. And that's something that's an area that we could be working in as well.
I think also to comment on what you said, I guess the whole psychiatric domain undergoes this transition slightly, right, into identifying more symptom-specific circuits, like in schizophrenia. I think you mentioned that before, someone else mentioned it. It's a very heterogeneous disease currently, but we could maybe not only help diagnose it better, but also redefine the concept of these diseases, similar to depression and so on. So I think there's a huge opportunity, but you've all been working on that anyways.
So I just wanted to follow up on that a little bit. I think all these diseases are very heterogeneous and very comorbid. And I think really two directions that we need to push forward are precision medicine, multimodal precision medicine, and also early and earlier detection, even from infants. And then we can do behavioral interventions and so on and so forth. If we get early biomarkers, and it can be multimodal of nature, then we can do a lot more for society in terms of being able to prevent the disorders from even happening. Or we can also have novel interventions, behavioral interventions, or pharmacological interventions like CBD or real-time fMRI, augmented, mindfulness augmented real-time fMRI feedback. Many different interventions that you could do early on that may be able to basically take the course away from its natural trajectory. So I think that's one way we should go.
I would also say, I agree with that, and I would also say not only multimodal, but multi-disorder, right? It is important because there are a lot of comorbidities. There are a lot of commonalities across these different disorders. If you start, I mean, we've started doing this in some of these larger data sets where we have schizophrenia, substance use, autism, ADHD, et cetera. And there are some really consistent things that you see across those. And there's also some divergent things, right? And so you don't look at that and you miss that important piece. And so, again, sort of crossing these different domains so we can kind of get a larger picture and get out of the silos. There's not been a lot of focus on generalized specificity, right? And looking, okay, I can predict this disease, but maybe that same thing predicts every other disease, right? So maybe it's not specific to that particular disorder.
So if there are people who want to talk about funding, first of all, what we're talking about here is actually similar to sort of RDoc's concept of there are axes which are not projected onto the DSM that people should look at for psychiatric disorders. But I guess from a funding priorities level, we need more work on how to actually combine incommensurate data. We need more theoretical work. I'm not smart enough to do it. A couple people in the room are smart enough to do it, but this is where our energy, I think, needs to be coming up with hardware to make multimodal stuff. People with engineering skills like myself, we can do that. I mean, they're straightforward challenges, but coming up with ways to combine eye movements with, photoplethysmograph data and make a prediction, that's actually really hard.
It's all hard, but we're all doing different things.
Could I suggest, so Joy, you and I had a great conversation, I believe, right after lunch, and this sort of leads into it, where we have sort of these silos that we all exist in. We're all particularly good, usually at one-ish things, maybe like 1.5 things. And the question comes up a lot about training, and the science didn't start with the t-test, and how oftentimes in, say, molecular neuroscience, all we teach people are t-tests. But how could we benefit future trainees in the field by giving them access? Like, how should training work to capitalize? Should it include multimodal imaging? Should it include more advanced statistics? Should it include stuff?
Are you asking me?
Well, I'm asking you, and then I figured other people would jump in.
Well, I kind of tend to think a little bit out of the box, but I think that students ought to be allowed to make more mistakes. I think that as one explores and one goes where no one has gone before, that we need to endorse the probability that we're not going to be successful. And that we learn a little bit from our Yankee ingenuity spirit, that we pick up the pieces and move on and learn from our mistakes. And I think in training people, people watch us. We have to do that in order to teach our students how to do that. But I do think that if we have this creativity embedded into our practices, I like what Mark was saying, we need to embed it into our priorities for funding, so that playfulness even is something that helps us do science. We don't talk about that very much, but exactly.
So I also think that at a certain point we cannot expect people to become more experts than the top level experts. And in a way that is what we're expecting from the students that we're currently training, that they're going to know more than what we know, or know more than what we know jointly. I think it's really important that we also train people on doing team science, working together. So different set of skills. So rather than going for the personal, individual incentives, which are of course very often linked to funding schemes, career opportunities, etc. I think we should also train people that they contribute to future society by working together, by being part of a team, possibly by being less visible as a middle author on a paper. But that is possibly more important than always going for the first or last authorship papers and going for the quick win. I think so. Open science, open data, and team science, they all relate to each other. They're all part of a new way that we're now, at least that I'm trying to contribute to as a culture in getting people to work together. And that's where I think that funding schemes should be aligned with this in changing the incentive structure such that the social aspect of working together is also really fun. But it should also be rewarded to have fun and to have the social interaction in the work.
Just regarding the siloing question, by the way, I completely endorse what you're saying. Several years ago, I'm proud to say I developed a training program whose purpose was to try to abstract the question of various imaging modalities into the question of what is measurement, what is finding a signal, what is forming, doing pattern analysis. And, such a thing is possible. And I think that training students to come to a more abstract level of what it is they're doing is extremely, extremely important. I mean, rather than teaching somebody in massive detail about how you build a gradient amplifier for an MR scanner, it's much more important, I think, to think with students about what it is you can do to a signal to make it detectable. And because those things are robust against the future when we either can't afford MRIs or they've become obsolete.
Related to what you're saying, the fact that you have different people teaming up together in the context of multimodal imaging, I guess very, very few people know as much as Robert about EEG and MEG, so he probably is the best person you would like to have in your team, and for fMRI, there's probably also a similar types of level of expertise. Do you think you can do like top-level research without the deep expertise in each of the modalities, or is it not plausible? I mean, can we do good science without fully understanding how MR acquisition works? Kind of provocative statement after what you said, Mark, about understanding how this.
Well, yes, because I mean, I don't really know how the internals of the ICA algorithm work, but I know how to get data in and out of it, and I have to, part of team science is to turn around and recognize that the person next to you has expertise superior to your own in something. And you have to be able to trust the quality and respect the quality of the work that's being done by the other people on your team. I can't possibly take ownership of, I'm an MRI expert. I don't know anything about RF. I really don't. And it's because it's a specialized area, and yet I believe I can still do quality work.
I've heard this question put another way recently. In a room full of engineers and people who focus on methodology, can we really ask the right questions if we don't have the depth of training in neuroscience or if we don't have clinical education? It's something I struggle with because I don't actually know very much about the brain, and my whole job is trying to measure its signals well and interpret them. So I think, the silos thing comes up again and again, and a training program that we're setting up now is taking our students who are going to be in methods-based labs and sending them away from us to just be exposed to what matters and where we're actually needed versus what can we get done. Because I think for me that's the bigger gap than understanding another methodology. It's having the wisdom to direct those methodologies that might be the limit in the training that I see.
I saw an article the other day about how companies all around the U.S. have hired thousands and thousands of data scientists, and now they're in the companies and they're like, I don't know what to, I don't know what to ask or what to do, right? They're very bright, they've got a lot of tools, but they're not experts in a particular domain.
So this is something I was discussing, I think, with Molly earlier, but on the issue of team science, there's, the talk about training and talk about how it's funded, but if you want to break silos, how many psychology departments in this country have someone whose first identification is a methodologist at the faculty level? And I would say the group of you up here is probably at universities that are more skewed to having that person in your departments than many others, and I suspect that's not even true for all of you here. That if you want to have that team science and you want that training, having someone embedded in psychology who obviously is passionate about psychology, but also, their focus isn't on brain system X, their focus is on how do I make better methods, is still exceptionally rare. across the country. And that's not about NIH funding, that's not about training, that's about at the university level, how do you create job openings and who do you hire?
I couldn't agree more. I also think the same applies to hospitals, so university hospitals that I think without a specialty training, for example, in neurology, you cannot really get a faculty position, at least in Germany, but I think it's similar here. But we would need these people, right, that do translational science at these institutions, have access to patients, have some interest, of course, in neurology, but then be able to have their own research program there and be more methods geeks. So I totally agree.
So maybe if I can comment real quick on this. So my answer to this would be, explain your problem in such a way that CS departments somewhere else can understand what problem you are trying to solve in order to catch attention from other types of scientists. And this is something that you tend to, I think we tend all to believe that we are more or less able to fix all our problems, provided we work hard enough. And I think it's a kind of a misconception that the question that people doing applied neuroscience, cognitive neuroscience, they can ask a brilliant question, and their value is in asking the question and probably not finding how to answer it. And I think we all tend to be valued by how much we answer the question, and I think the question can be more interesting, valuable, and let the others fix it if they are better at solving it.
I just want to follow up the question. I'm sorry, I don't know your name from the floor. Daniel, okay. It's not clear to me that this is a problem of departments as of institutions. And, you know, you don't have to be in the psychology department in order to interact with the methods person someplace else. And, you know, there are a lot of reasons the psychology departments do this kind of thing. For example, they have their teaching program in which they have to have somebody teach core course in psychology, and an MRI guy isn't going to do that. So, you know, maybe the answer has more to do with how institutions both work on projects across departments and how training programs go across departments.
I've had this discussion with a couple of department chairs, and me being an engineer in a school of medicine, I had this problem. MR physicist develops a bunch of pulse sequences, gets on dozens of grants as a co-investigator because he enabled that grant. Does he deserve tenure? I mean, that's been a problem with kind of method-driven people, especially if we don't have a core biological, hypothesis. I just develop algorithms and I enable a lot of people. I do a lot better, I do a lot better science than I could do myself by helping enable other people do the science. But that's a challenge on the institution level to recognize that.
And it goes back to the institution in the sense that on tenure committees, you'll have people who, on a university tenure committee, will have a group of people, many of whom don't understand the differences that people in the engineering profession, in the humanities or in the sciences, those idiosyncrasies of those professions. And then if people try to cross disciplines, then that becomes a real problem. And so it's really about almost educating people. And the problem is and I can say this without any guilt. If I offend the people around here, well, it's unfortunate. But it's the older faculty. It's the people who can't think out of the box. And I'm lucky because I did my training, it was multidisciplinary. When I did my training as an undergrad, multidisciplinary was the new black, okay? So I did that, and then we went back to silos. We never got out of the silos. We were trying to. We put one foot out the door and then went back in. And now we're trying to step out of the silos again, and we're back to that same problem. So I totally understand, because I was a biophysicist in a school of medicine for many, many years. And it's about educating people and talking clearly and learning to speak different languages and learning to communicate using language that the people in another discipline can understand. So it's communication. And so that, getting back to the topic of training, which was the original question, try to train students to be able to speak multiple languages or be prepared to engage with people in a different way to what they're used to doing when they're just in their own discipline. Well, that's what I tell myself as well. And then the other thing with student training is just to learn to be your own worst critic. Do you trust your data? I mean, I get a lot of people come to me asking questions about EEG data, and it's after the fact. And so I'm trying to do damage control, and they'll say, my data look weird. Well, how do they look weird? Tell me, give me something more to go on. But people haven't looked at the individual subject data. People haven't looked at, sometimes that happens in fMRI studies. I mean, why would you average data? Why would you add noise in if you've got some studies that are not technically adequate? So that's what I would like to push, that people be more detail oriented in the sense of those methods details and be more discerning about how they apply the methods. And then if their data look weird, they might understand why.
I'll say this is the 5-minute warning. But that was an excellent answer to the question. There was a book that came out about 2 years ago that talks about how corporations, when they're in a startup mode, do quite well when they have what they call linkage people or generalists, people who aren't necessarily all experts in different things. And so it is tempting in universities to always hire the most senior person, the most expert person in a particular field, because they likely have grants in their own as PIs, and they likely have a funding history and a publication history as first authors. But as you all have mentioned, there are these second, third, and fourth authors who are these more generalist people. And sometimes I personally worry that the students who are naturally gravitating towards those generalists, sort of good at many, many things, linking different things together, aren't getting the faculty jobs. And all of you sit in prominent departments. And certainly when I was at many different universities, my take, and I'm happy if anyone wants to dispute this, is that sometimes an ad will go out for, say, a methodologist, as Dan mentioned, but by the time the ad actually gets advertised or the search committee actually gets around to it, it ends up being, oh, we don't really want that. We want someone who's a lot like all the rest of us. So you have vision departments who say, oh, we really need to get out of vision. We should hire someone who does autism or hearing or taste. And then by the time the actual pool of candidates comes in, it ends up being only vision people at the end of the day.
So since we apparently have funding officers listening, I'll go and say something appropriately controversial. How many people in this room have grants from NSF? Anybody? And how many people have written them? All right, so a couple people. So a big difference between NIH and NSF grants is an NSF grant actually has a mandatory section about education. And we are responsible, in order to get an NSF grant, to talk about how we're going to go ahead and teach somebody something. And that isn't there in the NIH grants. Whether or not that's a good thing, that's really tough, because we have a population supporting the NIH, that is the American people, who are looking for more and more translational, direct approaches from my lab to my mom's care. But I think that the education component might want to be revisited.
I just wanted to speak to the issue that we raised just a couple of conversations before about people not getting the credit is due for being the interim person or the technical person. And this is such an old problem that we all understand it perfectly well, but there's not a lot done about it. But there are some actions that institutions can and sometimes do take. For example, I don't want to use Yale as a good example, but that's where I'm from. But I mean, so don't take this wrong, but once in a while, some institutions do things sort of right. And in this case, the institution has expanded the numbers of tracks that people can be promoted on. And so in addition to the conventional tenure track, which is more the first and the last author type track, there are tracks in research, there are tracks, there's the clinical investigator tracks, there's several other tracks. I sit on a tenure committee and I can't even keep track of them. But there are lots of ways to promote people for contributions that are collaborative. And the more this happens, the more these old guys on the tenure committee sort of teach each other. You know, most of them are trainable if you say things enough times. And eventually they kind of get it that these people are really valuable. So there's hope. I guess that's why I'm saying this, that there are ways to solve this problem, and the supporting of different tracks with different criteria is one of them. Well, we'll say it again. We'll repeat it. Yeah.
I guess we are getting near end of time today, but I wanted to bring this back to one other multimodal question comment that might continue into discussions the next 2 days. When we were talking about the different applications, it felt like there's this distinction between simultaneous multimodal and multimodal. And when someone says multimodal, kind of, at least in my mind and a lot of other people, I think not just me, the idea is it's simultaneous. And it seems like the applications there are epilepsy, consciousness, and maybe collecting the same data we would rather collect but in less total time. But this distinction between what we can do with non-simultaneous and what we can do in simultaneous seems to sort of be an undercurrent for a lot of discussions, but not quite as explicit. So I'm hoping maybe over the next 2 days, maybe a little bit in the last remaining minutes, we can talk about sort of where the biggest benefits for each are.
Nope. Okay, yeah. So when I hear multimodal, I think of it as a big umbrella, right? So I think of you know, I often categorize it as dimension matched or dimension mismatched, right? So sometimes you can link on subjects, you can link on time, you can link on space, you can link on different things, right? And there has to be something common, but it doesn't have to be time necessarily, right? I also would say that for things that are simultaneous, there are tons of applications to that, not just those three that you mentioned, right? So you know, as like Katie mentioned, the time-varying connectivity and sort of validating that and looking at relationships with sleep, arousal, all sorts of things there, but also disease. So I guess that's my position. I don't think, you know, maybe that could be communicated more clearly to folks, and especially if you start thinking about, you know, incorporating. There are lots of things that are asynchronous that are really valuable to have. You know, things like social media, things like you know, GPS stuff, and you know, environmental, macro environmental types of things that are also, again, multimodal and imaging to me are broad. Like I would say EEG is imaging, right? I don't know if everyone would, but anything that has a spatial component to it. So I think sort of broadly in that context.
I can, sure. So I actually, it's a very welcome question. I deleted that slide from my set because whenever I talk about multimodal imaging, this is one of the first questions I want to address is what's the significance of simultaneity? And there are a huge number of applications where it's essential, but not all of them. So let me start off with the things I think about. We think about spontaneous behavioral responses. We think about epilepsy. We think about sleep staging, where the tools that measure the phenomenon are different than the ones that you're using as your observational tools. We think about anything that's uncontrolled from a behavioral perspective is the kind of thing where you have to go to the simultaneous cases. If evoked responses, that's a whole different story. And if you're looking at simple evoked responses, you generally don't need the concurrency. And as Vince has pointed out, you can even retroactively enforce the concurrency of multiple time resolution timescales in proper analysis. But they are two different domains, and I completely respect your comment for that, as well as the others here, that multimodal at some level simply means collecting data from multiple different sources and combining them.
Yeah, just to add to that, I think certain studies in cognitive neuroscience and social neuroscience, you can only expose the subjects to the stimulus once. And that's mandatory to do a simultaneous investigation. That said, if you work in other areas where you think, and you've actually tried an experiment and you've done serial experiments and you know that the effects aren't going to change in a given subject, then why make things complicated? I would separate. I would want higher quality EEG. I would want higher quality fMRI. I don't want to kill myself and put the two together just because I can. And yeah, exactly. So that's one thing. The second thing is that some things don't lend themselves to combination. I'm going to talk on Friday about an intracranial EEG data set that that we're using, and we're using white matter tractography and white matter tract endpoints to inform us about the functional neurophysiology. So there are horses for courses, and I think, as Vince said, it's a broad umbrella. And so you have to think about the scientific question. Is it appropriate for the scientific question that we're asking? And then you choose the methods that you think are going to be optimal for that.
That brings us to time. I want to thank all of our panelists for participating today and all of our speakers, if we could give them a round of applause.